Hey everybody, this is Tom Salami. Welcome back to Device Talks Tuesdays. It's great to have you here. We had a little bit of a break while we put on Device Talks Boston. I hope you were able to join us there. If you missed out, don't miss out on Device Talks Minnesota. It's coming up. But we're here today to talk about updates to ISO 10993-1, focus on foreseeable misuse. Very happy to have our friends from Nelson Labs. It's a Cetera Health company here today. Nelson is a frequent contributor to Device Talks Tuesdays. Their sessions are always very popular and very informative. So I know I have light work today because they've got a lot of great things to talk about. But first, I'll go over the ground rules. We want your questions. Submit your questions in the Ask Question widget. Submit them if you're watching us live right now on May 20th at 4:01 p.m. Eastern. Submit them if you're watching this on demand. Those questions will go over to Nelson Labs. So we want your questions. Don't wait to ask them. If one comes to mind, type right in and get it in there so we know how many we have to deal with. If we're not able to handle them all during this precious 60-minute session, then your questions will be packed up and sent over to Nelson Labs, and they'll be able to follow up. So if you have a question and want an answer, you'll get it one way or the other. Just do your part and make sure you submit it in the question widget. Do not submit it in the attendee chat box. That's for us just to talk to amongst ourselves, share where we are, to make comments if we'd like. So let us know where you are right now. We'd love to know what the weather is. If you want to tell us what your favorite snack food is or some kind of icebreaker conversation, throw that in there as well. We'd love to learn about our attendees. So real live attendees, folks who are attending right now, you can help us get a gauge of what you're feeling and what you're thinking about by using our emojis. Yes, it sounds silly, but getting a thumbs up, smiley face, etc., is always nice to see while we're presenting, or while these fine folks are presenting. So be liberal with the buttons, share some love and some emojis, and make this an even more fun interactive experience. There we go. There's our first emoji. Thank you very much for the party emoji and the smile. There they are. If you go in the resource widget, you'll be able to download and walk away with this presentation. So make sure you do that and check that out. There we go. Look at that. That's fantastic. It's like a snowstorm of emojis going back up. It's fantastic. And yes, introduce yourself and interact with others on the chat box. But once again, please don't ask your questions there. We really need the questions to go through the Q&A box. If you do ask your questions there, I will publicly shame you and tell you to put it in the Q&A box. So I don't want to do that. You don't want that to happen. So let's just all try to get along. If you're having any troubleshooting problems, any kind of difficulties, the best thing to do is to refresh your browser. That'll take care of 99.999% of every problem. But if your problems persist and you haven't already done this, try closing your apps and browsers, your Zooms, and everything else that sucks all your memory and your energy. Get rid of that. If you still need help, there's that the little sandwich hamburger icon up there that can you can click that and get some help for troubleshooting tips. But of course, we'll always we're here to help you at all as well. So use the question widget. Just tell Amira Weiner, our producer, that you're having some trouble, and she'll reach out and make sure you're all set so you can hear everything and see everything. All right, so here's our Device Talks Podcast Network. More is coming out. Just subscribe in any major podcast player. You can find all of these at devicetalks.com as well. And I mentioned our conferences. Registration is open for Device Talks Minnesota, which is happening on June 11th. And planning is well underway for Device Talks West, our innovation industry forums. Those take place on October 15th and 16th at Santa Clara. Once again, you can register right now, and we'd love to see you in person. So we're here, though, to hear from the fine folks at Nelson Labs. Thanks again to Nelson Labs, a Cetera company, for sponsoring this episode of the Device Talks Tuesdays webinars. Click, and I'd love to introduce our speakers of the day. We have Lucas Ma. He's the biocompatibility expert at Nelson Labs. Robert Mueller, senior expert consultant toxicologist at Nelson Labs. And Katrina Hurst, biocompatibility expert at Nelson Labs. Katie, I saved you for last because I think you're leading us off, correct? I can hand the
Yes.
hand the microphone over to you. Take it away.
Okay, great. Thank you. Yeah, so I'm Dr. Katrina Hurst, and I'm one of the biocomp experts here at Nelson Labs. We also have presenting today Lucas Ma and Robert Mueller. So they're each biocomp or toxicologists. And today we're going to be talking about, so I'll start with just a handful of updates to ISO 10993. And then we're going to dive into one of those updates being foreseeable misuse. So diving into what exactly does that mean and what can that entail? How does that impact you or in your job and the devices that you're bringing to market in the future? So, sorry. So, one of the first things that you'll see when 10993 Part 1, the new one, is published, is that there's no longer this giant chart that outlines, here's all the different devices, here's all the endpoints that you need to address. Before there was very much a checkbox approach that could be taken, and that's gone. So if that was your method before, things are going to get a little more tricky for you. So now they're kind of split up. And so there's a table for intact skin contact, and then there's another table for mucosal membrane contact. And within each one of those, for example, there's the limited, long-term, prolonged, own table for breached surfaces or tissue contact, and then circulating blood has its own table as well. So now we have different tables for each of these different kind of tissues or organs or vessels, whatever the contact is. Another big thing that you're going to see is that the systemic toxicities have kind of just been broadened, right? Before you had acute, subacute, subchronic, chronic, material-mediated pyrogenicity, and they're all their own little checkbox. That's gone too. So yeah, it's definitely 10993-11 on acute systemic toxicity or on systemic toxicities is updating to kind of come in line with what's going on with Part 1. But again, these aren't official. At the point that the standard's at right now, there's not likely to be huge changes, but there is potential change still. So, take everything I say with a grain of salt. But so one other thing that I think is probably a big thing is this kind of removal of material-mediated pyrogenicity. So that's kind of been removed from that table. And now there's more discussion on when that actually needs to be assessed. So it really kind of takes it away for a lot of devices. So if your device is not novel materials, if it's something, ABS, polycarbonates, metals, right? These things that we're very familiar with that are used all the time in medical devices, you really aren't going to need that testing anymore. So there's definitely more discussion on how to make that clearer in the standard now. But the hope is that we'll be able to reduce that testing. Something else though, so we're stepping back from the MMP, but genotoxicity is more prevalent than it was before. So there were some devices, so for example, mucosal membrane contact genotoxicity, depending on the duration, may or may not have been needed, whereas it is more likely to be needed now. So if you haven't had to do it before, you might have to do it now with these updates. So those are kind of just like brief overview of endpoint changes. The rest are pretty much staying the same. The one thing, or the couple other things that are really going to impact, I think, more people or more devices is how contact is considered in the new Part 1. So before, if you had a device and it was used for 5 minutes and for 7 days, then we would go, oh, it's 35 minutes of contact, that's limited contact. So, we don't need to do these additional endpoints. But now the standard outlines more like that if it's being used for 7 days, regardless of the duration within that day, we're looking at 7 days of contact, or whatever the duration is.
And I think so it's worth mentioning.
Oh yeah, sorry, Rob.
Oh, not a problem. I think it's worth mentioning here that for those familiar with chemical characterization or toxicological risk assessments, that this way of categorizing the duration is how those standards have been categorizing the exposure duration for quite a while now. So it is a harmonization along with those standards.
Yeah, that's a really great point. And I think where this will come more into play is for devices with tissue contact, where it was just this brief contact for a few days. We didn't necessarily go into doing chemical characterization, and so maybe you haven't had to make that consideration before, but now we will. So that's a really great point. We're really, I would say in general, all the standards are really trying to be more harmonized. So one of the other points that kind of feeds into this that is more emphasized in the updated standard is bioaccumulation. And the exact definition isn't 100% clear just yet, but there is more discussion on considerations that you'll need to consider this potential, right? You're going to need to look more at your materials and not just what's on your device, but what's used during the manufacturing or even the packaging. Are there things that are potentially could bioaccumulate in the body, and how to address that? So there's definitely a lot more expertise that's going to be needed to really do these assessments. So that, which is also part of the update, is that you need to have someone who has that expertise to write these assessments. The last one on this slide is talking about, so previously the standard did talk about life cycle assessment, but it really didn't give very much guidance. So there's more guidance now on how to address that. I won't go into great detail because we want to jump into the next topic, but so there's more guidance, which is great, but there's still going to be kind of open-ended. And so needing to have someone with that expertise to help guide that. And there's also more on kind of what device degradation, when that can occur. So, before it was like, oh, my material doesn't degrade, so I don't need to talk about that. But now, it wants you to more or less prove that either that isn't going to happen with your device, whether that's with functional testing, chemical characterization. There's different ways that you can support whether your materials could degrade over the life of the device. So a lot more on that, more guidance on that as well. So the other things that are updated. So 1093 is always focused on risk assessment, like risk-based assessments. What's new is that it's bringing in the ISO 14971, which wasn't really there previously. And so what that is emphasizing, and there's several flow charts to kind of walk you through how to do this. But the basic is, what biological hazards can we identify, and what could be the harm, and then what does that mean as far as patient risk or likelihood of harm. So that's something that's new, even though to a certain degree it's always been there, but now we're actually bringing in that standard. And then the other thing that's more recent is, before the focus was very much on the patient, what's contacting the patient, how's it contacting the patient. So now there is a little bit more on, well, could this interact with the user, and how could it interact with the user? I mean, ultimately, And that user might not just be the surgeon that's wearing gloves. Who else could, and how could there be this potential risk to anyone really coming in contact with your device? And then the last thing is, and this is what we're really going to spend the rest of the time talking about, is these updates to assessing foreseeable misuse. Before you were like, this is how my device is used, this won't contact the patient, I don't have to address it. Well, now you need to think about, how could a user or a patient misuse my device, and what risk does that come, how does that risk come into play? So that's what we'll talk more about, and I'll hand it over to Lucas.
Great, thank you, Katie. So what is foreseeable misuse? Foreseeable misuse is when a medical device is used or may make contact in a way that may seem reasonable in a clinical setting, but it wasn't initially intended by the manufacturer. And it can be intentional or unintentional. What I mean by this is the user may, during a procedure, may choose to rest the device on the patient while performing something else, or they may be inserting it a little too aggressively or manipulating it in a way where parts of the device, which weren't considered contacting, are now making contact with skin or tissue. And unintentional would include things where, for a urinary bag, if the user is changing or draining it, they may raise it a little too high, causing some of the fluids in the tubing to flow back into the patient. This is misuse because the tubing, which was designed or intended only to transfer fluids away from the patient, is now acting as an indirect contacting component for fluids. Another example of misuse would be if a trocar with an inflatable balloon were to somehow rupture, maybe from another sharp device brushing against it, it's now exposing these internal areas to the tissue or fluid. And I do want to stress the foreseeable misuse part of the term, because misuse is a very broad term. An example I have of just misuse would be when I was notified of a time when a surgeon was taking foam packaging and using it as a pledget. So for those who are unfamiliar with what a pledget is, it is a small component that a suture is looped through in order to provide additional support and to prevent the suture from tearing the tissue when used. And essentially what this means is the surgeon had taken primary packaging and turned it into a long-term implant device. So this is just misuse. I would argue that it's not foreseeable misuse, because as a trained medical professional, I think many of them wouldn't, their first instinct wouldn't be to take packaging and turn it into an impromptu medical device. So we are focusing on things that are within just how it's still used in a clinical setting, but things where, again, there's issues with parts of the component touching the patient, the end user, or just used in ways that may not have been intended. So why should we consider it? So first and foremost, patient safety is always our top priority. And the reason why foreseeable misuse is so important is because there are potential areas of these devices that are now contacting the patient or the end user. And the FDA and other notified bodies have always considered this type of risk in their submissions. And that's why it is very important when you're creating your evaluation plan and thinking of all the test prep, that you do provide clear descriptions of your device, as well as clear instructions on how it would be used in a clinical setting. And the reason why I say this is because a lot of the questions I get are deficiencies regarding misuse. They stem from just a misunderstanding, or when the device information isn't properly provided to the reviewer. You need to remember that the reviewers will not be familiar with your device, especially if it's a brand new one and there's no predicate device. And so they'll ask things like, well, how do you know for sure that this handle doesn't contact the patient? How do you know for sure that only half of the shaft goes into the patient? What if this part were to brush up against another part of the device component, which is metal, causing particulates? So these are all things I've seen asked, and they get asked when, again, they're very unfamiliar with the device and may not fully understand how it's used. So when you create this clear narrative for the reviewer right off the bat, it helps them understand the full picture. And when you're actually presenting types of foreseeable misuse, they are much more likely to understand and agree with you as well. And so we can avoid all of these questions during the review. And going off of that is the test preparation. When it comes to preparing a sample, Part 12 does state that any kind of non-patient contact component should be excluded from the testing. So this is very straightforward. However, this becomes an issue for the testing is if something gets excluded, because as Katie said, you say, this isn't patient contact, there's no way it is, so we should just remove it. However, if the reviewer comes back and says, we don't agree with this approach, then you would have to retest it because it wasn't properly evaluated the first time around. An example I have of this is there was a ship clip that was to be removed from a device prior to use. It was considered non-contacting because it was just to protect the device during transfer. However, they found out on the field that sometimes when the surgeon was removing it, there were actually shavings of it remaining in the shaft, and which would then go into the patient. Therefore, we had to go back and actually do full testing on this material as well.
I also wanted to bring up,
yeah,
with that last one, sorry. So maybe you have a device that you intend to be used once, but the foreseeable misuse is that it could be used multiple times. So now we're going from a limited contact to, a prolonged or long-term, especially when we're considering the new kind of outlook on how, I say new, but not new, on how a device contact should be considered. So that's another way, so maybe it's not just what's contacting the patient, but how long is it contacting the patient?
Right. And that's a very good point too, because once you're changing from limited to the other prolonged or even long-term, there are so many other endpoints that we would now need to evaluate and potentially test for. Thanks, Katie. So now that we know what foreseeable misuse is and how we determine and why it's important, how do we determine what is considered reasonable foreseeable misuse? So you can't start off with just looking at any kind of data you have. If it's a device that's still in new product development, you can look at any kind of mechanical test data that you have. When I was working on an NPD project and coordinating all the testing, at a later point, while they were doing this stress test to determine just how many uses this device can go through, they were actually noticing particulate generation on the distal end of the device. So this wasn't something that was initially captured. So therefore, upon these observations, we now had to address particulate testing and make sure that what was generated, it wasn't being left in the patient. Basically, now that we had a potential of particulates, we had to see how we can mitigate this risk. And if there are predicate devices or something out on the field, or if you're doing any kind of clinical testing or clinical trials, you can get feedback from the surgeon as well, and just to see how it is being used. A lot of times, or just sometimes, surgeons may ask you, is it okay to use it in this manner, or how about for these procedures? If you find yourself getting the same questions from different parties over and over again, then that is something that you may need to consider. Even though it wasn't initially intended for it, if there are multiple people asking, then that is a potential chance of misuse. And other times it's just looking at how a device is, the types of procedures that you're thinking of, and just thinking what kind of issues can stem from just using it as is. And how do we address these? So ways would be to, again, provide clear information on how it's to be used, and maybe also updating your IFU if there are any other types of misuse that come up, to say, this is how it's supposed to be used, and then you add contraindications to say, this is definitely not intended for these types of procedures. And other ways is just implementing control measures. Again, going back to NPD, I've worked on projects where they've actually made physical design changes to prevent certain issues from coming up. They could be actual physical design changes to prevent the user themselves to use it or manipulate it in a certain way, thereby ensuring certain parts of the device do not come into contact with the end user or the patient. And if there are identified risks, and we have to evaluate these materials, then sometimes we do have to do testing and basically categorize these components or these devices accordingly based off of the misuse. So with that, I'll hand it off to Robert, who will give some specific examples from the toxicology side and how misuse is affected by that as well.
Yeah, as Lucas mentioned, if possible, it's advantageous to leverage existing data as opposed to generating brand new data for potential misuse. And that is especially true with chemical characterization, especially due to its high cost and long turnaround time. One of the key criteria for a chemical characterization study to be considered suitable for toxicological assessment is having a screening limit at or below the analytical evaluation threshold, or AET. I've provided the equation of the AET here from 10993 Part 18, the standard providing guidance on chemical characterization. To simplify the concept, the study must be sufficiently sensitive based upon a device categorization. The screening limit of the study must be at or below this AET. The AET is calculated from a dose-based threshold, the DBT, in the equation here. Most commonly, the toxicological threshold of concern, or TTC, which is in the numerator of the equation. The appropriate TTC is based on duration of use, and as the duration increases, the TTC value decreases. Seen here in the TTC table taken from ISO 21726. Common misuse consideration, as has been mentioned, includes repeated exposure or extended exposure duration, such as a device being left in the patient. A chromatogram taken from ISO 10993 Part 18 is here on this slide to give you an example of the AET versus a screening limit. If the recalculated AET from the device misuse categorization is below the analytical screening limit, chemicals that should have been detected, identified, and quantified were missed, and the study would not be considered suitable for toxicological assessment. Another potential misuse consideration with a direct impact on the AET is the maximum number of devices clinically used. If a misuse scenario results in more devices being used, the AET would need to be recalculated. In the AET equation, the maximum daily clinical use of devices is the term C, located in the denominator of the term in the numerator. An increase in number of devices results in a lower calculated AET, which could again result in a chemical characterization study whose screening limit is not at or below the AET and would therefore not be sufficiently sensitive enough for a toxicological assessment.
So, Robert, I just want to bring up the previous slide as well, because you showed that if it's not properly evaluated, there are certain parts that would go missing, and that would potentially result in a retest. So just on average, how long does a chemical characterization test along with the toxicological risk assessment
that's a great question. So right now, we can anticipate a chemical characterization study lasting from about 12 to 18 weeks. That does not include the subsequent toxicological risk assessment, which is typically 4 to 6 weeks after that. So it's quite a substantial amount of time if you're needing to redo the testing.
Right. Thanks, Robert.
So not only would the chemical characterization need to be reevaluated for its suitability from the misuse categorization, but in addition, the previously performed toxicological risk assessment would also need to be reevaluated. Tolerable intakes to which exposures are compared for safety are calculated from the available data addressing specific biological endpoints. A change in duration of exposure may result in additional endpoints being required for assessment, such as from a subchronic duration now to chronic toxicity, or needing to address genotoxicity. The toxicological risk assessment would need to reevaluate if the selected points of departure adequately address these additional biological endpoints. Next, a tolerable intake is specifically calculated for the clinical route of exposure. If misuse considers a different route, so let's say oral versus inhalation versus parenteral, than initially assessed, the toxicological assessment would need to be updated to address this change. And finally, ISO 10993 Part 17, or the standard that provides guidance on how to perform a toxicological risk assessment, prescribes uncertainty factors for calculating the tolerable intake based upon toxicological information, its gaps, or extrapolations, and for the quality of the point of departure. These values would need to be adjusted as appropriate to calculate the final tolerable intake. If the existing studies performed do not address the nature of misuse, a common consideration that is often asked of me is if it's possible to perform a paper toxicological assessment. Such a paper assessment is often considered for new chemical residuals from, say, use of a cleaning wipe on the device that wasn't previously considered or cleared in the IFU, inadvertent generation of particulate, or chemicals exposed from internal compartments now being exposed to the patient. The hurdles with this approach are twofold. The identity of the chemical must be known, and this is not just at the material level, but all of the constituents of this chemical, including impurities. And in addition, we need to know how much is being exposed to the patient. So the twofold points needed are identities and concentrations to be able to perform a paper toxicological assessment that in the end would really only be able to, generally speaking, be applied to systemic toxicity endpoints.
And Robert, I just wanted to point out regarding paper assessments that I've had people treat paper assessments kind of like the silver bullet of the tox assessments, just like how people would treat cytotoxicity as the silver bullet for biocom testing. A lot of times they ask, "Oh, we've had this change. Can we mitigate the risk with a cytotox?" I've had people ask, "Oh, can we mitigate this potential change or these leachables or identified compounds with a paper assessment?" And we're not saying no right off the bat, but there is just a lot of legwork and a lot of information that we would still need in order to explore this route. And it's not always a guaranteed success.
Yeah, I would say in general, when we're trying to address something with just a written assessment, we have to have a lot of information to support the approach we're taking, and a lot of literature to support that approach as well. Even though X, Y, and Z makes sense, if we don't have a lot of support for it, then it's probably going to get rejected. And again, like Lucas and Robert said, there's always that chance that the reviewer will be like, well, okay, and just not follow that logic, or not just, they don't want to take that risk, and they'll have you test anyways. So it's always, weighing your options and understanding what's your regulatory risk there as well.
Exactly. And I think this feeds in perfectly to our first case study example. So a sponsor came to us at Nelson Labs, and they had this fun device. It's a limited contacting external communicating device. They felt and identified for themselves and their company that there was a potential hazard, that if the physician was using this device with other metal surgical tools, roughly, they felt that potentially they could crack the device and now expose the patient to this chemical that was otherwise internal and would never contact the patient. So they approached the toxicological team and said, hey, we have this harm or this potential hazard of this internal chemical reaching the patient. Can you perform a paper assessment on this to help us understand what the overall impact is on patient safety? So the follow-up question was like we discussed in this last slide. So what is this chemical? What's in this chemical? They were able to provide us portions of what was in it, but there was a large number or percentage of the chemical that was proprietary, where they couldn't gather information. So for the known chemical constituents, we were able to actually identify what the harm to the patient was. But for the unknown constituents, we weren't able to really state what that possibly could be. In addition, because they really didn't have a way of testing how much was coming out or could reach the patient, we had to make an assumption that a worst case total amount of this chemical could come out of the device and reach the patient. With an unknown harm and quite a large concentration of this chemical that could potentially, in a worst case, reach the patient, the paper assessment was not favorable. We couldn't say that this was safe based upon the information that we had available.
So what they ended up doing from there was really, they had a bunch of testing in cadavers. So not necessarily clinical data, but they had data on functionality to show, they were really trying to break this device, and it wasn't actually happening. And so what they ended up having to do was use that to support that, really, even though there was this hazard and potential harm, the actual risk to the patient for it to happen was extremely low, almost zero or zero. So there's, more than one way to assess this potential misuse than just doing testing, right? So I think that's important to understand, that, testing isn't always the answer.
Mm-hmm. Right. Especially in this case, when they couldn't emulate any kind of, or have this actually occur during use, I would say that this wouldn't be foreseeable misuse, because, and to go along the route of, okay, well, let's evaluate it anyway, that wouldn't be reasonable.
And I think that's some of the real power in the new updated standard, right, is that they provide us a framework when it's based upon this risk assessment of really identifying what really truly needs to be assessed to understand patient safety versus something that really just isn't of concern.
Right, exactly. And I think the other thing that kind of, for everyone, this is one example, and we'll go through a couple more where the answers were different. But having, so like I talked about this degradation potential for life cycle. So having this testing, this functional testing, to show that your device isn't wearing or the surface is inert. And so that comes into play again when we're talking about what's the hazard, what's the harm. And so it's really, all these updates are really just focused on making sure that patient safety is truly addressed. Let's hop to the next one, I think. I'll talk about this one. So we had an example where there's these tubings that are used for dialysis treatments, and they come with caps on the tubing that are intended to be discarded. And then the tubing's used, the patient's connected to the dialysis machine, they go through their cycle, and then the tubing's removed, no contact with the cap. But there's that stint in the patient that the cap could be placed on. So then we go from no contact to now having long-term indirect circulating blood contact, which opens a huge avenue of testing. So, how can we address this misuse, right? So I would say in this case, and this maybe feeds into some of the questions I've seen on the chat, is, there is the potential to have these like warning, do not use, have it very clear that this cap is intended to be discarded and not used or reused at any point.
Mm-hmm. And Katie, I also wanted to add just another example is I mentioned design changes and things like that to help mitigate these risks of misuse. I've had teams actually change the colorant on these labels or things that are designed to be removed, just to make it even easier for them to see and just increase the visibility of these things that are meant to be discarded and not used within the procedure.
That's a really great example, because not everyone that's using these might read your big warning, even if it's like really big.
Right, exactly.
But yeah, doing something like that, having a discard on the actual portion or something like that, those are things that could help to remove that foreseeable misuse concern.
Right.
Do we have anything? I can't remember if there's anything else on this one. The last example that we had was for a device that was placed underneath the patient during surgery to kind of act as a neutralizer for electrical activity, if the device is being used electricity. And the manufacturer was like, well, it's on their back, it's opposite of the surgical site, and we don't foresee any breached or compromised contact, and this would only be intact skin contact. And then, when they were under review, it's like, well, what if the patient's on their side and this is placed, or what if the surgeon places this closer to the site, and there was just too many questions of what if, like Lucas brought up earlier, what if this, so then they had to go from being considered intact skin contact to now breached or tissue contact. So then they did end up having to do additional testing. So this is kind of one of those situations where, we kind of went through 3 examples where the answer for one was, doing functional testing. The answer for the second one was, making sure that labeling and use instructions, et cetera, are very clear. And then in this case, we couldn't really say, no, that'll never happen. So they did end up having to do additional testing. I think those are all the case studies that we had to share to kind of give different examples of different ways to approach foreseeable misuse. So we'll actually open up to the questions.
All right. Sounds great. get our Q&A session here. So we've got a bunch of questions here from folks. Thanks for your interest in the topic. We'll get to as many of them as we can in the 15 minutes that we have. Once again, if we don't get to your question, and we may not, please know that it will go to Nelson Labs and that they'll get back to you privately. And if you want one that's not read aloud, but you want to ask it privately or answer it privately, indicate that in the question, and I won't even bring it up. So some of the earlier questions are earlier on just sort of centered around where are we with approval? Is there a date in mind? What's the process here? Where are we? Where are we headed? What's likely to happen?
Yeah. So yeah, let's kind of. So it's technically kind of in the final stages of approval. I know this last week there was a lot of discussion on the new -1, and so there's potential that it could go to vote and be rejected, and then it basically goes back to point A. So, if that happens, then we're talking years down the road.
Okay.
Yeah, but if it goes to vote in the next few months and it passes, there's enough that are in favor with the updates. There are things that we've identified that need work. And if those are at least to the point where the logic is usable, then and it passes, then we're looking at it being sometime this year or very early next year.
All right. So Adam wins the first question of the webinar award, so we'll get to his first. What is the relationship between biocompatibility and toxicology? When does a biocomp assessment push into a toxicology assessment?
Yeah, that's a good question.
Robert, do you want to take this?
Yeah. So typically, right, toxicology is a tool within the 10993 frame. It is taking the information that is known about a chemical and using that information and comparing it to a tolerable intake or a derived tolerable intake for assessing exposure to this tolerable intake for determining safety. And it's able to be used for specific biological endpoints. Generally speaking, those endpoints are the systemic toxicity endpoints. So your subacute, subchronic, chronic, carcinogenicity, gene tox, etc. So while it's able to be used for those things like cytotox, and within the US and with the FDA, sensitization and irritation are not able to be addressed through a tox assessment.
All right. Rolling up, let's see. From Hassan, would the manufacturer be off the hook to evaluate for additional risks in endpoints if they contraindicate for specific misuse, even though we may know that a misuse is systemic? At what point do we draw a line between addressing foreseeable misuse during a biological evaluation and not getting dragged into accounting for off-label use by physicians, which is supposedly a non-regulated practice? That's a very well-crafted question, Hassan, and very well typed too, not a single typo, so thank you for that. But that was the easy part. On the answer part, any thoughts from our esteemed panel?
Yeah, I mean, so this is where this foreseeable misuse in the update is really tricky in addressing, because if it really is a foreseeable misuse, there is a really good chance, depending on the regulatory body and your reviewer, that even if you have this, don't use this for this or that, or only use once, or whatever the case may be, if the reviewer considers that the risk of that misuse is high enough, regardless of that, then you are very potentially looking at having to address it. And then there's also, depending on the device and how it's used, there's that potential, like Lucas brought up, where you do some kind of design change that would prevent that misuse. And that's something you can do to hopefully prevent that. But then outside of that, it honestly, and this is where it's going to be hard, is it'll probably come down to the reviewer and their approach and opinion on the topic. I wish I had a clear answer, but I don't.
Yeah.
Well, yeah. And I think it's that definition of foreseeable, right? You know, if you know, I think it was interesting in the question where they talked about systemic misuse. If something is all the time being misused and you're aware of that, that is a foreseeable misuse versus, I think, you know, Lucas's example again of someone using packaging in a very bizarre way in a surgery. Yeah. I would imagine that someone using packaging in surgery like that is very uncommon and not systemic. So that's not foreseeable and would not need to be addressed.
Right. So unfortunately, it really does come down to, it depends. And in the biocomp world, that's something that we always say. And in this case, it's no different, unfortunately, because it really depends on the actual type and the extremity of the misuse. Is it actually reasonable? For example, if you, again, going back to a device that I helped evaluate, it wasn't intended for neonates now. And so, of course, it specifically said that in the IFU. And so is that something that they would have to say, or would that be something that they would still have to evaluate for? Well, in that context, I would argue no, because the device itself was basically 3 feet long. And there was absolutely no way this could physically actually enter in a neonate. So again, it comes down to the context of how your device is used, as well as the potential misuse that is being brought up.
Okay, got a question here from Zoya. What would you suggest for balancing potential misuse versus ensuring a clinically representative test article for your testing? For example, a clinician rests a portion of a device on the patient during procedure, which isn't intended for patient contact. Would you include that portion within your testing at the risk of the rest of the device extract being diluted out with lower risk components?
Yeah, I'll take this one. So, yeah, that's interesting. So there are some things like, if it does rest on the patient, is that part of the patient draped? You know, if they're going to be draped, then really even though it's resting on the patient, it's not contacting them directly. But so to kind of go into, you know, if you see that foreseeable misuse and that really is directly contacting the patient, you know, do we include that portion in the testing? And I would say potentially yes, but what will be different is, so something that -12 outlines, and part of this was in the earlier portion that Lucas was talking about, is that those non-contacting, as far as intention, can be included in the test article, but then the surface area for those portions would not be included in the calculation for the volume of extraction. So we're not diluting the other components that way, but we're still including this potential, you know, misuse. And in that case, even so, if you have a device that's externally communicating limited, so you know you're doing cyto, irritation, sensitization, acute systemic tox, and potentially MMP. So really, if it's resting on the outside of the patient, on their arm or something, then you'd really only be looking at those first 3 tests, including that portion of the device, but not the acute systemic tox or MMP. So those are all things that, you know, which is why it's really important to have that biological evaluation plan, because it will outline, you know, this is the foreseeable misuse. So we're going to address that contact, for example. And this is how we're going to address it, is by including this portion in these tests without including the surface area. So we're still being very conservative. So that's kind of, I would say, you know, if it is something like that specific example, that's probably the approach that I would take.
All right. Question here from Robert. Could you address need to repeat testing when or if there is a new supplier of critical raw materials? Discuss equivalent but not identical specifications of materials.
It depends.
Okay. All right, next question then. No.
Yeah. I'll just say any device change needs to be addressed through a biological risk assessment, and depending on information available, etc., the conclusion may be additional testing, repeat testing, no testing. The conclusion can vary widely depending on the change and the use of the device.
All right. Let's see. I was scrolling around and I had a question here. It is from Johan. Is chemical characterization and toxicological assessment generally expected by the FDA for limited contact devices that come into contact with circulating blood, such as catheters?
So
Go
typically,
ahead, Lucas.
yeah. So typically for limited contacting devices with only systemic endpoints like that, you can address those through a combination of in vitro and in vivo tests. Usually, as of now, chemical characterization is used to address the prolonged or the long-term endpoints.
Great.
Yeah, so more or less, no, not necessarily. We have seen them ask for chemical characterization depending on the potential repeat use or something like that. So that's kind of where that misuse may come into play.
Okay. I've got a couple of questions here. One from Mark, one from Jasmine, kind of along the same lines. Mark asks, is there a rule of thumb for probability of harm for something to be considered, quote unquote, reasonably foreseeable misuse? For example, 1 in a million, if we can see it happening once across the entire life cycle of the product from launch to sunsetting, does that mean it's reasonably foreseeable? And Jasmine had I think kind of a similar question asking, would foreseeable misuse, is there a certain number of instances of misuse we need to assess? I'm wondering how far you need to go in finding foreseeable misuse. It seems to be, it's kind of an open-ended question.
Yeah.
What's foreseeable and what's not? I mean, any guidance?
Yeah, no, I mean, gosh, I wish there was a magic number that we could pick and it would be the cutoff. That would be super nice. Yeah, as of now, that I would say is not super clear, and that may be something that causes the new -1 to go back to draft, is that we need more guidance on that. So yeah, unfortunately there isn't super clear guidance, and so yeah, I don't have a real answer for that question, unfortunately. But yeah, just FYI, it may be something that comes down to the regulatory body and what they consider, if it is something that is a really high harm but a super low probability, in general, I would say it won't be severely under scrutiny. They want you to address it somehow, whether that's through showing the probability as being extremely low or some other form. There's something about identifying the misuse and then how you can address it. And it will vary depending on what that misuse is.
All right. And final question from Lindsay, because we'll wrap up. She asks, and it's kind of just a question, kind of a head scratching. I wouldn't expect the FDA to be publishing any new guidance on how to interpret the future update to ISO 10993-1. How is a manufacturer expected to make heads or tails of these important changes to expectations for testing? It sounds a little some frustration on her part, and understandable. Yeah,
definitely understandably so, and that's where, again, there's definitely debate on whether these updates will be fully accepted, and which countries will fully accept or only partially accept. Like there's a lot of potential for your submission to different countries having to be very different.
All right. Well, the bell has rung, so I think it's time for us to close out the webinar. There are a lot of questions here, a lot of really great questions here, very detailed questions here. So thank you to our Device Talks listeners, for our viewers, for taking part in such an active part in this conversation. As I said, all of these questions will be packed in a box and sent to Nelson Labs, so I'm sure they'll reach out. I'm seeing now folks are giving some emojis. Yes, please give our panel some emoji love for the conversation we've had today. I know there's a lot of uncertainty out there. Folks like Nelson Labs can certainly help, and again, they'll reach out to you individually if you have asked a question. With that, I want to thank our panel for joining us today and for leading this important conversation. Thanks for being here.
Great, thanks so much.
Great, and
thanks
thanks to and thanks to Nelson Labs for sponsoring this event. It's always great to have them as part of Device Talks Tuesday. And thanks, of course, to everyone out there who registered, who watched this live, who's now watching on demand. We really value you as part of our Device Talks community, and I'm really grateful you chose to spend some time with us today. So have a wonderful rest of your day, whether it's a Device Talks Tuesday or another day at your convenience that you're watching this. Take care, everybody.
Thank you.
Bye.